factors. And the raw materials for this process are in your ovaries or your testes, depending on you know what it depends on. They're diploid cells called either primary oocytes or primary spermatocytes, depending on what kind of gamete they make. Men produce sperm, you may have heard, and they produce it throughout their adult lives, whereas women are born with a certain amount of eggs that they'll release over many years after puberty. Here you might want to go watch the previous episode about mitosis again, because that's where we go into detail about each stage of the process. Once you're done with that, we can start making some baby makers. Now, just like with mitosis, there's a spell between rounds of cell division where the cell is gearing up for the next big split, and this is called interphase, when all the key players are replicating themselves. The long strings of DNA in the nucleus begin to duplicate, leaving 2 copies of every strand. To jog your memory about how DNA does this, we did a whole episode on it. You can watch it and come back. A similar process takes place with the centrosomes, a set of protein cylinders next to the nucleus that will regulate how all of the materials will be moved around along these ropey proteins called microtubules. And that brings us to the first round of meiosis, prophase 1. This is nearly the same as in mitosis. The centrosomes start heading to their corners of the cell, unspooling the microtubules, and the DNA clumps up with some proteins into chromosomes. Each single chromosome is linked to its duplicate copy to make an X-shaped double chromosome. Now keep this in mind, once attached, each single chromosome is called a chromatid, one on each side of the X. Each double chromosome has 2 chromatids. Here in meiosis, prophase 1 includes 2 additional and very important steps: crossover and homologous recombination. Remember that the point here is to end up with 4 sex cells that each have just 1 single chromosome from each of the homologous pairs.